Okay, good morning, everyone. Hopefully you're staying fairly dry and it wasn't too leaky in your places last night from all the rain. First thing I check is how many drips I have inside that place, you know. Just a little bit of an announcement here. Also, by the way, it was a good and bad sports day. Sharks won in overtime, so I was a little excited about that. Giants won, but unfortunately a bunch of LA teams also won, so so it was good and bad for me. But at least the Sharks did win in overtime, so that was fun. It's only the playoffs that count anyway, right? Of course, I sent out an email. Did you guys get the B-Space email that the office hour Thursdays, 2 to 3, like one person was showing up, so I stopped those. And talking to a bunch of you suggested having office hours right after class on Friday as a potential time, and also the room was open, so I grabbed it. So we'll have office hours 9:15 to 10. That's a.m., not p.m. Okay, I forgot to put that in the B-Space announcement. I assumed you guys thought about it. Then I realized I probably get to sleep a lot earlier than you guys do. So it is a.m. right afterwards. So it's starting today. So I'll have office hours right after class today. And today we're going to finish with female follicle development, menstrual cycle hormones, get into fertilization, pregnancy, and twins. One of my favorite topics, because I do have a set of twins. And I know there's at least 1 or 2 sets of twins out here. Maybe some of you guys have siblings that are twins. So we'll talk about that. And then if we get that far, pregnancy and labor hormones. Okay. Something I'm going to leave on the board during our discussion of the female reproductive system is something we talked about on Wednesday, and that is the male hormone control system. It is continuous due to a negative feedback. So the idea is to keep dihydrotestosterone at relatively constant levels, because that's what affects the spermatogenic cells. The hypothalamus kicks out GnRH, or secretes it, gonadotropin-releasing hormone, binds to its receptors in the anterior pituitary. The anterior pituitary response is to produce LH, luteinizing hormone, and follicle-stimulating hormone. Again, to reiterate something I mentioned Wednesday, those hormones were first discovered in females. Same hormones are in males, therefore they didn't change the name. Thank goodness for that. Okay? So that's men do not have follicles, nor do we have a corpus luteum, but we do have LH and FSH, luteinizing hormone and follicle-stimulating hormone. There's a bunch of other hormones in this system. I'm just giving you the basic pathway here. Luteinizing hormone, LH, binds to its receptor in the Leydig cells, converts cholesterol to testosterone. Keep that reaction in mind, because females do exactly the same thing in the fecal cells. The testosterone shoots across the basement membrane. Sertoli cells convert testosterone to dihydrotestosterone, and it's dihydrotestosterone that stimulates spermatogenesis, the spermatogenic cells. Testosterone comes out in the system. It has a negative feedback on the anterior pituitary and the hypothalamus. So if there's too much going on, testosterone levels go high, shut down LH, FSH, and GnRH, shuts down the system. If it gets too low, synthesis kicks back on, and testosterone levels come up. So again, testosterone levels go up and down, but they stay within a certain window of acceptability, which is different for every person. But there's on the average of 1 million people, you can get rough numbers here. Okay? Questions on that? It's continuous. Keep this in mind, because in females, it is not continuous. It's a very different system based on a lot of the same players, though there's differences, of course. Okay, so you should understand similarities and differences. We talked a little bit about seminiferous tubules, epididymis, vas deferens, seminal vesicles, prostate gland, bulbourethra gland, urethra. Know what's going on there. In terms of male erections, it's a nerve impulse. You get nitric oxide. Do a few steps. You activate an enzyme called guanylate cyclase. Again, you've had adenylate cyclase. This is guanylate cyclase. Critical not to mix up the two. It converts GTP to cyclic GMP. It is cyclic GMP through stuff you don't need to know, but it affects phosphorylation, ion channels, et cetera, et cetera, et cetera. Causes muscle relaxation and arterial dilation. One thing we did emphasize a little bit is phosphodiesterase shuts down or breaks down cyclic GMP, so it eliminates the response. And Viagra is an enzyme inhibitor of that particular phosphodiesterase. And there was a question in the back, I don't think the person's here right now, about temperature and spermatozoa. And I looked up a few studies. It turns out 37 degrees centigrade, which you should all recognize as body temperature, spermatozoa don't last very long. Okay, they get viability gets shut down, motility gets shut down. That is why men keep spermatozoa away from the body initially to maintain viability and motility, and that does it by reduced temperature. Okay, so just 37 degrees, they don't last that long, so it's an interesting reproductive factoid there. Okay, any questions on that? All right, so now to compare the DNA changes in spermatozoa and oogonia, you start out with these diploid cells, 46 chromosomes, 2C DNA. They undergo what I call a meiotic duplication. It can also be called a mitotic amplification. The initial step in mitosis and meiosis is virtually identical. Doubling of the DNA, condensation, the chromosomes condense. I call it, as some other people call it, meiotic duplication to enforce the concept that these cells are on the way to meiosis. There's no choice. A primary spermatocyte, once you make that, there's no choice. It continues in the meiotic process. So it's the same thing. Sometimes it's written as mitotic amplification or meiotic duplication. It's exactly the same process. Okay? Does everybody see that? So I'm emphasizing the meiotic pathway here. Basically, you go to 46 4C chromosomes. Males is pretty straightforward. First meiotic division, secondary spermatocytes, 23 chromosomes, 2C DNA. Second meiotic division, spermatids, which is the first true haploid that you see. And then spermatozoa, which is the gamete. So a spermatid is not the gamete. It has the gamete DNA, but is not the gamete. The gamete is technically the spermatozoa. Females, on the other hand, have a slightly different process here. Oogonia, through this meiotic duplication, form primary oocytes. Females are born with all the primary oocytes you're ever going to need. By puberty, there's about 400,000. The first meiotic division occurs at ovulation. We'll emphasize that a bit today, what that means. And you form a secondary oocyte and a polar body. I had made a mistake on the board in that I wrote this first polar body as 23 chromosome cDNA, but is incorrect. The first polar body is 23 chromosomes 2C DNA, because it's the first meiotic division. It's correct in the reader, and I wrote too fast on the board when I put that in. But at ovulation, the polar body that forms at ovulation is 23 chromosomes 2C DNA. Because of unequal cytokinesis, the secondary oocyte gets all the goodies, gets all the cytoplasm. The polar body is basically a nucleus, and it's gotten rid of. It's not functional, except in the movie Twins, where Danny DeVito came from the polar body, and Arnold Schwarzenegger came from the secondary oocyte. That's kind of the premise behind that movie. Next, most of the time, females produce secondary oocytes only upon fertilization. Entry of the spermatozoa nucleus, which we'll talk about today in more detail, slightly more detail. You form the ovum nucleus and another polar body. This polar body is 23 chromosome cDNA. So you make two polar bodies, differences in DNA. The ovum nucleus is a haploid gamete type DNA content. And then as you'll see, what happens is the spermatozoa nucleus, the DNA combines with the ovum nucleus, and eventually you make a zygote. We'll talk about that directly today. Questions?
Does the polar body just disintegrate, or does it have some sort of function?
Does the polar body disintegrate or have some sort of function? It has no known function. It doesn't. It gets degraded, or in the case of the one that gets ovulated, a lot of times it just gets sloughed off with the secondary oocyte. So females make a potential ovum every 28 days. But what you are making is a secondary oocyte, which is not the same thing as an ovum. The only time you see the gamete true haploid DNA content is after fertilization. Okay? Does everybody see that? So that's why sometimes generically the whole process is called eggs, because they're so different at different parts. Okay, questions on that? We'll get in. You'll see this again in a slightly different point of view. All right, questions? All right, good. So we started out with ovum development. In the ovary, at the beginning of the menstrual cycle, there's multiple. You're sitting there with 400,000 primary follicles. Follicles in the ovary with primary oocytes. That's why it's called a primary follicle. 20 or so start developing. And for reasons that are very poorly understood, even now, 1, sometimes 2, emerge as the winner. And they're the ones that start really developing. Okay? And that's called the primary follicle that develops. And as we talked about, through proliferation, estrogen and LH and FSH, which will become clear in the next 10 minutes or so, you have this primary oocyte surrounded by a membrane called the zona pellucida. And that's very important. And we'll talk why that's important. It has spermatozoa receptors, and which allows for the proper species of spermatozoa to enter the egg, to enter this in the first place. Surrounded by granulosa cells. Granulosa cells go from testosterone to estrogen. So these guys are producing tons of estrogen, which is then surrounded by theca cells, which convert cholesterol into testosterone. Okay? So this is what starts forming near the surface of the ovary. The one that wins out is that one. That is in case you have fraternal twins, we'll talk about 2 of them win out. But in this case, we're going to talk about 1. Okay? So what happens at that point is a bunch of fluid builds up around the primary oocyte. So here's this primary oocyte sitting in the ovary, and a bunch of fluid builds up. This is the fluid, and this is called the antrum. And there's a physical dynamic going on here. If you took a balloon and kept pumping water into it, my kids love to do this, what happens eventually if you don't stop putting water into it? It bursts, right? This fluid buildup allows for much easier bursting of the membrane of this ovary, the lining of the ovary, to push out the oocyte. So it builds up pressure around it, not unlike pumping water into a balloon. It's by analogy only. But you get a sense here that the physical dynamics are changing. Then what happens, you have a surge in LH, which in the females, just like the males, comes from the anterior pituitary. And you'll see where a little bit of this. This surge in LH causes ovulation, kicking this out. But in addition, you get the meiotic division. So kind of, I'm not too worried about the order of events here, but kind of in this process, you get the surge in LH, signals for ovulation. At the same time, you're signaling for the meiotic division. So it kind of all happens around the same time frame. And what happens is, what you pull out of this ovary is now a secondary oocyte. Sometimes a few granulosa cells comes with it. Does everybody see that? What's left? What is the primary oocyte surrounded with? What cell types is it surrounded with here at this stage? Granulosa cells and theca cells, correct? What happens is what's left, the theca cells plus the granulosa cells differentiate. Got to spell that right, granulosa. They differentiate into the corpus luteum. It's a differentiation process. This is the corpus luteum here. So the part of the ovary that's left after you kick out the secondary oocyte is the corpus luteum. The important thing about the corpus luteum is it makes estrogen plus progesterone. And that's really important in the menstrual cycle and female reproductive hormonal control. So the result of this surge in LH is ovulation, the first meiotic division, and the formation of the corpus luteum in the ovary. exactly where that primary oocyte was sitting, where the theca cells and the granulosa cells were, and they come from those cells. And it makes a new steroid in this system, and that is the progesterone. Okay? And then, after this gets kicked out, this secondary oocyte goes to the, we'll talk about this in about 10 minutes, goes to the fallopian tubes, maybe 20 minutes. Goes to the fallopian tubes, where fertilization takes place, and you get the second meiotic division. And we'll talk about blastocyst formation, probably in about 20 minutes. Okay? Questions on this point? It's very important to see this. So reproductively, what steroids are being made here? If I said you do it in this, up to post-ovulation in the ovary, what are the different steroids that are made by females? Testosterone, okay, which is immediately converted into estrogen. And what else gets made? Progesterone. Very good. So the steroids that you see stably or at the end of the day is really estrogen and progesterone. Because all the testosterone, if everything is normal aromatase activities, always gets converted into estrogen. Okay, the female normal reproductive cycles has extraordinarily little testosterone as it gets out into the system. Okay? So, yes? Pardon?
What does progesterone use for?
What is progesterone used for? We will get to that. That will become hopefully very clear in about 10 minutes. It's a very important part of the reproductive cycle. Now, all this comes together in what we call the menstrual cycle. And we'll just put this up here. See if there should be room here. Involving a couple different entities. Yeah, this will fit. Which on the average in a female is about 28 days. And we'll put ovulation right in the middle here. In the first, and it's about 14 days on either side of this. And the first 14 days is called the follicular phase. And that is because that is the development of the primary follicle. Very straightforward to think about. The second 14 days is called the luteal phase in the ovary. And that is because during the last 14 days, you have the presence of the corpus luteum, which sticks around from ovulation till the end of the roughly the end of the menstrual cycle. Okay? For the steroids, hormones, basically estrogen comes up. This is estrogen, shoots up, down a little bit and stays pretty high. Progesterone is pretty low until after ovulation. And that is because you only have the corpus luteum formed after ovulation. And that is the only part of this that makes progesterone under these situations. Non-pregnancy situations. Okay? And then you have the anterior pituitary hormones. What you have is, you'll see how this happens in a second, you get a spike in LH, or the LH surge, and you get a smaller spike in FSH. And you'll see how that happens. It is the surge in LH that kicks on and triggers ovulation. And you'll see why that happens. It has to do with the type of feedback. In females, as you'll see, you shift from negative to positive feedback during the menstrual cycle. And that causes the surge in LH. All right? Then, down in the endometrial, then this bottom graph is the endometrial lining. Endometrium lining. Endometrium of the uterus, which is the lining in the uterus. And this is thickness. And this is a very important part of this whole system, too, because you can ovulate all you want, but if what you make, the blastocyst, if it does not attach to the uterine lining or the endometrium, you're never going to get pregnant, or at least the pregnancy that maintains itself. Okay? So this thickness, the first part of this Thickness goes down the first 3 or 4 days. This is the menstrual flow. And then it builds up and builds up past ovulation. Optimal thickness about 7 days after ovulation, which is really nice timing, because from the time of fertilization, which occurs right after ovulation generally, to when it wants to attach to the endometrial lining and in a blastocyst stage takes about 7 to 8 days to form. So it's all timed together that the lining of the endometrium isn't optimal till about 7 days after ovulation, which is exactly when you want it optimal. Okay, any questions on that? Does everybody see that? This is what's going on in the uterus lining. This is the ovary up here. Okay, questions? Yes? The question is, why can't some women get pregnant? There's multiple reasons. Everything from not properly developing the primary follicle, and we're going to talk about fertility drugs, or briefly mention it in a second, all the way to the endometrium lining doesn't work right. And attachment doesn't occur. There's almost any reason you can think of probably occurs in someone. All right? It's very important. It's a very complicated process in the sense there's lots of steps to it. Okay? Now this first part of this cycle here in the endometrium cycle is called the proliferative phase, and the second part is called the secretory phase. Because what's happening after the menstrual flow is you proliferate more of the lining cells, and the secretory phase you're getting it ready for the attachment of the blastocyst. Okay? So the point here is hormonal differences. Estrogen is up before ovulation. After ovulation, you have estrogen plus progesterone. And right before ovulation, you have a surge in LH. So how does all that take place? And now we're going to, I'm going to go into the hormonal control mechanisms here. Okay? And I want you to, during this discussion, contrast and compare it to what I'm going to leave up here with the male hormonal system. So a lot of the players are the same. So initially, my first diagram is going to be from about here to about here on the menstrual cycle. First 7 days or so, although this will differ from person to person. So you start out with the hypothalamus. And it produces GnRH, gonadotropin releasing hormone. Don't need to spell it out every time. It should be in the reader. Binds to receptors in the anterior pituitary. Okay. And the anterior pituitary produces LH and FSH, luteinizing hormone and follicle stimulating hormone. What is the difference between what I wrote on the board to what's going on with the males? Nothing. Correct answer. Okay? LH, early on in the menstrual cycle, causes the theca cells, binds to receptors in the theca cells to convert cholesterol into testosterone. Note the similarity to what theca cells in females do to what Leydig cells in the males do. Identical reaction response to the identical hormone. But there are not Leydig cells in females. It's theca cells. And there's slightly more to it, but not too much more. FSH causes through aromatase its receptor, causes testosterone conversion into estrogen, and estrogen stimulates the follicle development, one of the main players in follicle development and proliferation. Okay, so the idea here is to make estrogen. In the beginning of this cycle, you don't have that many granulosa cells compared to what you have later on. At these lower levels of estrogen, and I'll put this board back up here, at the lower levels of estrogen, or lower or lowest estrogen, you've got your classic negative feedback system. The system is trying to maintain the steady state levels of estrogen. So you have GnRH making FSH, LH. You produce estrogen, which makes the granulosa cells grow. Okay? And these are the granulosa cells here. Very important to put this in. They're the ones that convert testosterone into estrogen. You make the estrogen, you try negative feedback. Too much of it, bring it down. Too little, bring it back up. Everybody see that? That's what's happening in the first 6 or 7 days. Yes?
When would you have low estrogen?
Pardon?
When would you have low estrogen?
To a relative to any one person, you have low estrogen early in the cycle, when you don't have a lot of granulosa cells. Okay? It differs for every person. And I should say not low, but lower is probably the correct term. So I'll put this here. This is a very important concept. This is time. This is estrogen levels. What happens is, as the granulosa cells proliferate, one of the responses to estrogen is more proliferation. As the granulosa cells proliferate, the system makes more and more estrogen. So you go up like this in estrogen levels. And for any one individual, there's a threshold where below it, it's considered lowish or lower estrogen versus high estrogen, which occurs part of the way through the cycle. So that's this transition for each person is different, but occurs roughly here. Okay? The reason is the granulosa cells, the response to estrogen is to proliferate. So if you have more granulosa cells, guess what? You have more estrogen. So your estrogen levels are going up prior to ovulation. Okay, any questions? Because I'm going to complicate this in a second here in a very important way. Yeah. Pardon?
Is there an inflection point in that line, or is that just
What, this line?
Yeah.
This is a threshold, which differs from every individual. This?
Yeah.
What do you mean by inflection point? It gradually goes up, crosses some threshold that's different for every individual, and keeps going up. Okay, this is a theoretical threshold. All right? That level is different. For some women, what you call low, see, the reason I'm stressing this, the hormonal mechanism of response is different depending if it's low or high estrogen. For some women, this threshold is happening here. For other women, it's happening here. Okay? So why am I bringing this out? At the high levels of estrogen, as soon as you cross that threshold, you no longer have negative feedback. What you have is a positive feedback. So at the high levels of estrogen, so this is lowish estrogen, so eventually you make a lot of estrogen. This is estrogen at very high levels. Once you make it at high levels, I'll put the board back down. At the high levels, instead of a negative feedback, you now have a positive feedback. What is the difference between negative and positive feedback? Pardon?
Positive feedback amplifies the...
Right. In positive feedback, you amplify the change, right? Negative feedback, you want to keep things constant. So once a female crosses that threshold, you now have high levels of estrogen and you have positive feedback. As a result, instead of turning and dialing down or wanting to dial down the estrogen, what do you want to do? You dial it up. And when you dial it up, what is the response? You make a ton of LH and FSH, and that causes the surge in LH, that positive feedback. The result of the positive feedback is, in fact, this surge in LH and a smaller surge in FSH. It is due to positive feedback. So when a female, crosses this threshold, which is different for every individual, you shift from negative cooperativity. So at low estrogen, you've got negative, feedback, not cooperativity. And when you go to high levels, you have positive feedback. So and therefore, instead of keeping LH and FSH lowish and constant, you stimulate LH. And this LH, this surge, causes the ovulation and the first meiotic division. Does everybody see that? Question here first.
Is it possible that the luteinizing hormone is
Which one?
the LH goes up first before the FSH because
No. What happens is estrogen positively stimulates a massive production in LH. It is estrogen that stimulates this.
before the estrogen goes high, does the luteinizing hormone is there more testosterone? Should there be more testosterone?
Yeah, okay. When you give this positive feedback, you got to make more testosterone because you're making more estrogen. But it's due not so much to how much testosterone you make, it's due to the fact you have many more granulosa and theca cells around. Okay, you're proliferating. It's like having this classroom, and all of a sudden, by the end of the lecture, you proliferate, and there's 5,000 seats, and another 4,000 students show up. In that case, you might as well go to the Greek theater in the rain, right? But the point is, you have more cells there. Okay, there's a question around here somewhere, right here.
How does one break out of the negative feedback?
Oh, you'll see. How does one break out of the negative feedback? Nobody knows exactly what this means to cross the threshold. It is my opinion that the response of the estrogen receptors is different. And it's not clear exactly what it means. Some people think you need a little bit higher levels of estrogen for the receptors to have primarily, say, protein-protein versus protein-DNA interactions. There's speculation out there. There's nothing that's unequivocally been shown to be the reason for that. But it's the change in the response. Obviously, the anterior pituitary is changing its response due to the concentration effect. Okay, there's a question here.
So can we say the positive feedback portion is like the next 7 days in the cycle?
Approximately. Where this threshold occurs is different for every individual. But it's somewhere in here. It's prior to the surge in LH. That's the important thing, because it causes the surge in LH.
And like how many days is the cycle that's between like when we switch to positive feedback and we get the LH surge?
Again, it's a very good question. The answer is different for every person. What's the timing? It's different for every person. It's days, probably days. But that's not important as much as knowing before ovulation. This is all going on pre-ovulation. Because at ovulation, it's a very good question. At ovulation, what is the new hormone during ovulation that we haven't talked about yet? Progesterone. Aha. Something important there. At ovulation, the corpus luteum is now in play. So you have the corpus luteum, and here you have the hypothalamus, the anterior pituitary, the theca cells, and the granulosa cells. I'm giving this all shorthand here. So you've got this massive positive feedback going on, right? Because you're making all this estrogen, correct? But at ovulation, what is the corpus luteum making? Estrogen plus progesterone. Estrogen plus progesterone, adding the second steroid, wipes out the entire feedback system and has this massive inhibition of both the anterior pituitary and the hypothalamus. So in the presence of estrogen plus progesterone, you wipe out GnRH production, you wipe out LH and FSH production, and you dampen the entire and inhibit the entire system. That's what goes on vis-a-vis the corpus luteum. Okay? Yes?
So like in the case of combination birth control pills, this is why we don't ovulate?
Thank you. That was exactly my next point. Great segue. The question is, in this system, what would make a great female contraceptive? Estrogen plus progesterone. So what you're doing is, in female contraceptives, is causing this inhibitory effect to stay. And therefore, you dampen GnRH, you dampen LH and FSH, you don't ovulate, and therefore no chance of pregnancy. Okay? Now, given this, everybody see that? And the reason why you can take it as a pill as opposed to an injection is these are steroids. They get through the stomach, they're absorbed, and everybody's fine. As in contrast to insulin, diabetics have to take it by a needle because if you ate insulin, you'd convert it into amino acids in your stomach. So it's a steroid, so you can take it. Now, given this system, what makes a good fertility drug in general, theoretically? Keep going. Keep thinking about the system. Okay, you want to make estrogen, right? You don't want to give people estrogen because that increases risk for breast cancer and uterine cancers and all this kind of stuff. What would make a good fertility drug? Keep going. Upstream. LH, FSH, and GnRH. A lot of the fertility drugs are based on enhancing or providing versions of GnRH, LH, and FSH. And as a result, when the follicles develop, instead of one developing, a lot of times multiple ones develop. So that's why it's very common, not common, but happens a fair amount, that women who take fertility drugs can in fact have multiple follicles develop and therefore fraternal twins. Okay? Or in that case, twins. A lot of times it's triplets and quadruplets, etc. All right? Any questions on that? Does everybody see this? Yes? What causes what? Well, it's not so much unequal. Not every hormone is always produced at the same level. Physiologically, you just make more LH than FSH. It's as simple as that. Okay? It's not what's the word for it? You don't have to. You're probably making excess anyway, so you don't need to worry about how much you make. Secondly, there's probably more receptors for LH than FSH too. But it works. FSH is clearly working because FSH stimulates the production of estrogen from testosterone. And normal females have plenty of estrogen. So it's definitely working. Okay? Good question. All right. So we're going to talk very briefly about fertilization and get back into some of this. In fertilization, what happens is the spermatozoa finds the zona, this is the ZP, or the zona pellucida. Just one brief comments here. And these guys have sperm receptors in it. The contact of the spermatozoa is random. It's a random contact. But the sperm receptors allow for more stable attachment once it occurs. And once this takes place, there's a lot of changes in the cell. So I want to give you a sense why this kicks a little bit of the context of kicking on the meiotic division. When the spermatozoa hits this, the acrosome, the enzymes are released. Here's the acrosome. Releases enzymes that breaks down the zona pellucida. ZP here is zona pellucida. When all this starts taking place, you have an increase in respiration in the cell. You've got calcium waves going on. You've got ion channels opening and closing, etc. A lot of metabolic events. And in this context of cellular turmoil, you got the meiotic division of the secondary oocyte nucleus. Okay? Continue this down here. So then what happens during this turmoil is you got membrane fusion and the nucleus of the spermatozoa gets kicked in. What is the DNA content of this nucleus of the spermatozoa? If I said to you, spermatozoa nucleus DNA content, what's your answer? All right, 23 chromosomes, C DNA. Very good. C is the correct answer because I asked for DNA content, but chromosome is 23C. During this context, you've got the secondary oocyte nucleus sitting around. What's the DNA content of that? 23-2C, right? Of the secondary. After fertilization, you have the meiotic division, And now you have the ovum nucleus, which is what? 23C, as is the spermatozoa. And you also have the polar body that gets kicked out. And this is the 23C polar body. Okay? So during fertilization, you've got respiration, ion channels, calcium waves, just complete turmoil. Sort of how I felt when the Sharks won the game last night in that overtime goal. Okay? So as a result of this, you got this meiotic division taking place, and you got the ovum nucleus. So then what happens is, we'll do this over here, is you got the sperm nucleus, the ovum nucleus, the membrane, the nuclear membranes break up. So now you have the DNA for the spermatozoa, and you have the DNA for the ovum. Okay? What's the DNA content of this DNA? Just the spermatozoa DNA? 23C. It hasn't changed yet. Inside the cell, you actually have the full complement of a diploid DNA, but it's not diploid yet. The DNA then recombines. You form now a nuclear envelope or nuclear membrane. And at that point in time, you've got the nucleus in this cell, 46 chromosomes, 2C DNA. It's now diploid. This is called a zygote. Okay, does everybody see that? Upon fertilization, you get this turmoil going on. In addition to this turmoil, you also strengthen the zona pellucida, called the cortical reaction. Let me just put this over here. You strengthen the membrane. And you have a cortical reaction. So what happens is you have these vesicles. The spermatozoa comes in. These vesicles come up and change the texture of the zona pellucida. Strengthens it so you do not have polyspermy. So you don't have another sperm coming in once this original sperm brings in its nuclei. So it's a good fail-safe mechanism there. Okay? A good beginning of a sci-fi story, perhaps. They have an extra nuclear, trisomy, and get an extra set of chromosomes and everything. Okay? All this takes place. You get this meiotic division. The ovum nucleus, the DNA, the membranes break apart. The DNA sits around for a second, momentarily recombines. The nuclear membrane reforms, and now you have a zygote. Everybody see that? As you can imagine, a lot can go wrong. And does sometimes. Yes? Oh, these are the cortical reaction. These are cortical vesicles. Okay, and these come up. They have different content in the zona pellucida. Comes up, changes the lipid, carbohydrate, oligosaccharide environment, some proteins and so forth. Mixture of stuff is now different. And therefore, I think it gets rid of the sperm receptors also. A couple of things going on there. And this membrane is now different, so you don't have extra sperm attached. Or extra sperm bringing in their nucleus. Okay? It's all part of what goes on. Questions? Okay, so in the last 7 or 8 minutes here, we're going to go over, I'll leave that up there for now. We're going to go over, in a slightly bigger picture, what happens upon where it all takes place. So here's the ovary. Let me get my other chalk. Here's the ovary. And you kick out, what do you ovulate? What type of egg, quote unquote, do you ovulate? A secondary oocyte, right. Primary is wrong. Ovum's wrong. You make a secondary oocyte. This gets picked up by the fallopian tubes. And when it is in the fallopian tubes where you get fertilization in most cases. Okay? And the first thing after fertilization is a bunch of cleavage reactions. So what are those? I can do this over here. You have a bunch of cleavage reactions. What happens is the zygote that we formed over here, you get a 2-cell stage, a 4-cell stage, and if I counted right, an 8-cell stage. And it's called a ball of cells or a morula. I think it's, as I remember, the Latin term for mulberry. If you can picture a mulberry, you get a sense of what this morula looks like. You have 8 cells. These 8 cells are completely indistinguishable. What happens in these cleavage reactions is you're splitting the cytoplasm, you're dividing the nucleus and splitting the cytoplasm. They're all diploid, obviously. But it doesn't get, each cell's smaller than the next stage of division, up to the 8-cell morula stage. I point this out for 2 reasons. Identical twins form by breaking up of these cells. So these are all genetically identical. They are all, as we'll talk about Monday, totipotent stem cells. Any one of those cells up to the 8-cell stage, you can break one off, form an entire individual out of it. You can break 2 off and form 2 individuals. You can have identical quadruplets that they break off multiple times. As long as you're within this 8-cell stage, you're absolutely identical. Okay? And this is going on here, these cleavage reactions. And then you get something called compaction. And this, what compaction is, these 8 cells just come together much more tightly. And at this point in time, they're probably no longer considered, they're genetically identical, but what that happens afterwards is not as clear-cut identical. Okay? Keep this in mind, because this is where when we talk about stem cells. Up to this point takes about 3 to 4 days. Think about the endometrial lining. This is taking place right here. When is the optimal thickness of the endometrial lining? What's the optimal thickness time, approximately? 7 days. We're only at 4 days. So what happens is, the next 4 days, next couple days, and this is the endometrium lining here, in the uterus. Within the next, by 7 or 8 days, you form something called a blastocyst. Part of the blastocyst has fluid called a blastocele. This is a fluid buildup. The other part has got cells called the inner cell mass. And as we'll talk about with stem cells, this is the primary choice of getting embryonic stem cells. It's coming from the inner cell mass of the blastocyst. Okay? Soon after that, this blastocyst implants into the endometrium lining. And that's occurring right here at day 7 where it's optimal. And let me put this right up here. In the last minute or 2 here. What we'll leave you with is when it implants. So we're going to do a blow up of this area here. The part that implants is called the trophoblast. This is the part of the blastocyst that connects up with the endometrium lining cells. And you start forming the placenta, which is part maternal, part fetal. What the placenta does, one of the first things that happens is the placenta starts making a hormone called HCG. Human chorionic gonadotropin. What this does is the human HCG's role is to maintain the corpus luteum, because you want to keep estrogen plus progesterone around, right, at that point in time for the next 9 months or so. You only keep it around for the first 3 months. So the placenta makes a hormone, HCG. We'll get into the details of this on Monday, but just one point I want to make is two minor points. The basis of the pregnancy test is the production of HCG from the placenta. Other than a tumor, the only time you really see HCG being made is right at the beginning, from the placenta, at the beginning of pregnancy. So at day 7 after fertilization, loosely, you can pick up a little bit of HCG, or day 8. Secondly, some sports players will take HCG to help build up muscle mass and recover from injury. Manny Ramirez was known for that. Those guys follow baseball, took HCG. Because in males, it has effects on muscle development and in response to injuries. Okay? So you are your hormones. Have a good weekend. We'll pick this up on Monday.